Hello, everybody. Welcome to today's webinar.
My name is Jaymar. I'm a Senior Marketing Associate here at Nanion Technologies, and I'm glad to be your host for today's session. Today, we will be talking about unlocking the full potential of your SURFE data with my colleague and scientific expert, Dr. Andre Bazzone. He will be telling us more about advanced techniques to get the most out of your SURFE data. So joining us today as the moderator is my colleague, Dr. Maria Barmes. She's the product manager of the SURFE systems here at Nanion Technologies. And Maria will start with a short introduction and will be later moderating the Q&A session. My pleasure then to leave you with Maria.
Thank you, Jaymar. And thank you, Andre, and thank you, everybody, for participating. So we are very excited to have our first internal webinar on a purely technical content since a long time. Usually our webinars host scientific speakers from the community, and this time we do it differently and present something ourselves. Please give us feedback if you want. So we would be really happy to hear if that is something we should do more frequently, if that is appreciated. So we're happy to hear your feedback at any time. I just want to emphasize, please approach us if you have follow-up questions, if your questions cannot be answered during the webinar, if you have too little time, or if you have more advanced questions or technical questions that you want to discuss with us directly, just approach us. I guess you have our contacts anyhow. I also highlight here the support email address. That's always good to use because there's always somebody responding. Then before we start, I just want to do a last-minute invitation to our Nanion user meeting. You can also participate remotely in the user meeting. So it's end of October, 28th, 29th. So we're very happy if many SURFE users are attending. And this year we have the first time a user workshop in advance of the meeting program, where we just talk about SURFE topics. It's a little workshop with practical topics. We have a news and notable session, also a practical part about the data analysis, referencing to today's webinar. We have time for networking and talking between users, and we'll look into stoichiometry assays and some housekeeping of your SURFE N1 instruments. So we would be very happy to have many users joining. That workshop, of course, will be in person only. Now, without further ado, I want to hand over to Andre.
Thanks a lot, Maria. So you should be able to see my screen now.
Yes.
And perfect. Welcome, everyone. Thank you for joining. So before straight up starting with the advanced analyzing techniques, I want to highlight why we actually selected that topic. So every one of you knows the basics of SSM-based electrophysiology, I guess. So just briefly, substance concentration jumps trigger the protein activity. And if the protein activity is electrogenic, we measure a capacitive current. The good thing is every current measured is a result of the substance provided via the solution exchange. But the challenge is that every current measured is a transient current. So how can you actually find out what this transient current actually reflects? Currents could have so many different origins, resulting from the target protein or non-specific interactions of the substance with the membrane, or even other components on the sensor. Current from the target protein can reflect transport, can reflect binding or conformational transitions. And non-specific currents may involve binding to the membrane surface, insertion of the substances into the membrane, a substance leak, or interactions with non-target proteins. And Well, the fun is, in many cases, your current reflects a good combination of several causes. And here, with that webinar, we want to provide guidance in how to tackle these challenges and, well, how to identify what is actually going on. And I actually should put that a little bit more positive. SSME enables access to so many information. You just need to understand how you can dissect the information of interest. And that's the overall goal of this session here. I divided that into 3 sections. Here is a brief overview. First of all, we will have a look on how to distinguish the different types of currents from each other, assuming there is only one type of current that is measured under the conditions used. Then we will have a look at more complicated situations where multiple currents of different origins overlay. Here, I will focus on 5 different methods, how you can separate the current contributions. A common example is a fast pre-steady state current measured when the substrate binds to the protein, followed by a slower transport phase. But of course, also artifacts could overlay with a current. And lastly, I will highlight some approaches on how to figure out what the different currents actually mean in terms of reactions in the transport cycle, or in other words, how to map those currents to molecular reactions within the protein. Let's start with non-specific currents. We often call those artifacts. How can you find out if artifacts are present? I can't stress this too often. Controls are very important. Control samples have the same lipid and protein background, but lack the protein of interest. For example, empty liposomes, if you're working with proteoliposomes, or when you work with membranes from an overexpressing cell line, you can use membranes from a parental cell line that didn't overexpress the target protein. Here are 2 examples. On the top, there is TMEM175, a cation channel. On the left-hand side, you see that we measured potassium flux by potassium concentration jumps, and we observed positive currents as expected. The control, however, on the right-hand side, revealed large negative currents. Those are also present in the sample currents, but we would never know without measuring a control. The second example on the bottom is a galactonate transporter. Currents with the sample show negative artifacts preceding the actual transporter current. But of course, without the control, those could have been attributed to a pre-steady state phase coming from the transporter itself. So what is the mechanism behind the most artifacts? It is ion binding and unbinding to the membrane surface. This is concentration dependent and typically very fast. When you see a slow current component, in addition to the fast spike at the beginning, like you see for proton jumps, that's the figure on the bottom right, this indicates an additional leak pathway. Another artifact mechanism is related to aromatic or lipophilic substances. Here it is about a slower insertion of the substance between lipid head groups or into the membrane interior. I want to highlight one recent publication, I think, from 2025, where the substance membrane interaction was studied using SSME. So yes, there was no interest in protein function in that study. It's just the membrane substance interaction which was studied. So it's actually an own research field. And the substances used were both charged and aromatic. So as a consequence, you get a biphasic current, a fast one, and a slower one, which reflects the insertion process. Here at the bottom, you see typical artifact currents representing fast ion binding on the left, and slow insertion of compounds into the membrane on the right. One important characteristic is the symmetry of the peaks between on and off currents. What binds also unbinds with similar kinetics because both is about surface effects. In contrast, transport currents depend on the kinetic properties of the target protein and are affected by the type of driving force. And the upper two figures show actual transport currents. At the time point of the off signal, when the substrates are washed out of the sensor, the driving force is mainly the voltage that was generated by the target protein during the transport phase. The on signal instead was only affected by the substrate gradient as provided with the activating solution. And as a consequence, typically off currents are reduced in amplitude 2 to 4-fold and also decaying more slowly compared to the on currents. The integral between on and off currents, however, is the same. So what goes in must go out, unless the protein works asymmetric, of course. So this asymmetric behavior of transport currents, in contrast to artifact currents, are a very good indicator to distinguish those types of currents. To get an idea which concentrations are critical for what type of substance, we have a table in the SURFE manual where we show which type of chemicals are more prone to generating artifacts. For example, you will see that metals generate artifacts at much lower concentrations compared to monovalent cations. This was it about binding artifacts. Now there are also more rare scenarios where the activity of the protein of other proteins interfere with the assay. Again, very rare compared to the artifacts resulting from membrane interaction, but still. One recent example is the lysosomal calcium channel TRPML1. We knew calcium generates artifacts by membrane interaction, but the amplitude that was left after TRPML inhibition using MLSI3 on the top left was just too high to just reflect calcium membrane interaction. Employing a general calcium channel blocker, ruthenium red on the top right, further reduced the current. So we indeed had contamination of plasma membrane, and these plasma membrane calcium channels activity overlaid with our TRPML1 assay. So we knew what we had to do. We had to improve the sample purity. Another interesting phenomenon here is when working with membranes from overexpressing cell lines. And indeed, in the control, there could be a basal activity of the target protein if it's not knocked out. So you need to consider that as well if you want to compare controls with your samples. So what if you see artifacts? What can you do? There are 3 different strategies. First, sample development to increase the signal of the target protein beyond the artifact amplitude. Second, assay development to reduce the artifact fraction of the signal. And if those 2 are out of question and the artifact is still above, let's say, 10 to 20% of the total signal, then you should consider subtracting artifact currents recorded with controls from the sample currents. And here you can subtract peak currents or integrals directly to correct your dose response curve. Typically, as you can see, the top right figure is a very nice example, actually. Typically, this reduces the EC50 because artifact currents dose response is more linear. And well, that was one possibility. The second possibility, if you have very small assay windows, if the artifacts are pretty close in amplitude compared to the actual transporter signal, then you can try to average current traces from different sensors and subtract average traces from each other. The advantage here is that this keeps distinct current features alive, allowing for better data interpretation and analysis afterwards. Now, bridging from artifacts to actual target-specific currents. How can you distinguish, for example, a transport from a pre-steady state current? So one first indication is that the kinetics in most cases is pretty different. Pre-steady state currents are typically fast. Transport currents usually decay more slowly. But this is not a full story. It's not always true. And well, also you should remind that concept. I want to highlight two exceptions from that. For example, transport currents mediated by channels can be even faster than ion binding to the membrane. As you see in the top figure, the blue curve is a channel, and the dark red is actual ion binding. And the second exception, there are pre-steady state reactions in transporters that might be even slower than charging processes mediated by transport. So very, very slow binding interactions, so to say. And that's at the bottom example. This is a glucose-induced transition in the proton-carbon sugar transporter xyl-E. However, what you can do is looking at the symmetry between on and off currents. Again, the symmetry between on and off currents provides assistance if it is more of a surface effect or a full translocation across the membrane. What most researchers use as a proof for transport activity is the dependence of the kinetics, of the decay kinetics on the lipid to protein ratio. For a pre-steady state current, the decay time is a consequence of the kinetic properties of the underlying molecular process. It is independent on how many proteins are measured. So at higher protein densities, only the peak and integral values increase, as shown in the upper figure, but the current shape is unaffected. The current shape reflects the kinetic rate of the measured reaction. For a transport current, the lower figure, the decay phase is a result of the charging process mediated by the actual transport. The more proteins are inside a membrane, the faster the membrane charges, the faster the equilibrium is reached, and the faster the current decays. So these principles have been used in literature to prove that people observe real transport in SSME recordings. Higher LPR resulting in broader currents means the current reflects the transport. On the other hand, when the LPR leaves the current shape unaffected, this is viewed as a proof for a pre-steady state current. As shown here, for the slow conformational transition induced by glucose binding to xyl-E, which looks almost identical to the transport current triggered by xylose, the main substrate. Only with the LPR dependence, we could reveal that the glucose is only a binder but is not transported. And that is also confirmed by radioactive transport assays. And below it is shown that the recorded current for an electroneutral sodium proton exchanger reflects only half of the transport cycle, classical pre-steady state current, independent of LPR. And also, again, a very nice recent publication used the LPR dependence of the peak shape to distinguish substrate which only bind from those which are actually transported. LPR variation has one major problem. It can only be easily applied for proteoliposons. So if you work with membranes, that's more difficult to achieve. However, besides LPR, there are three different approaches you can take. The first one, when you are looking at secondary active transporters, then play around with concentrations and gradients of co-substrates. Adding or removing driving forces can reveal distinct current shapes, indicative of additional pre-steady state or transport phases. So if you look at those traces presented here, you see under certain conditions, the peak shape gets broader. And that means under those conditions, where you have the additional driving forces, you actually see transport. And what you saw before is only a pre-steady state phase. Second thing you can try is applying modulators, for example, blockers that specifically block transport phases, but leave the binding of the substrate unaffected. That has been done in literature. And last but not least, you can also compare transport deficient mutants or different transporter variants with different substrate specificity to correlate certain current features with pre-steady state currents and others with the transport current. And also for that, there are a vast amount of examples in literature. Now, assuming the current is more complex, what are the methods you can use to characterize individual current components? First, but most importantly, use your eyes. Most things you see are real. So this is about the visual check. What do you need to look for? First, pay attention to kinks in the current rise or decay. Pay attention also to currents of different directionalities or even multiple peaks in the trace, like double or even triple peaks. What can help a lot is comparing current shapes with 3 mm and 1 mm sensors. 1 mm sensors improve the time resolution and therefore improve the separation of fast and slow current components. The consequence will be differences in signal shape when pre-steady state and transport currents overlap. For example, at the bottom, you see that when switching from 3 mm to 1 mm sensors, the kink switches from the current rise to the current decay phase because the pre-steady state phase is accelerated. And on the other hand, if the current shape is not affected by the sensor type, it is unlikely that the current components, that there are multiple current components underlying the measured current, as shown here for PEP-T1. Next, exponential fittings. There is, again, a vast amount of information about the different exponential decay properties of currents measured with transporters. And this slide should just show you that, well, there is a lot of information hidden in the shape of the current, and those can be characterized via exponential fittings, of course. So exponential fitting might even help if a visual check fails, if you don't see visually multiple components. One example is shown here. The current better fits to a bi-exponential than a mono-exponential function, indicating the existence of two current components. You might think, well, more variables, better fitting, but if you look at the right figure, if you vary the experimental conditions for that transporter, the existence of a negative current phase is revealed, and that actually could explain the deviation from the mono-exponential fit. But of course, there are more obvious scenarios. Here you can directly compare fit quality with mono or bi-exponential fittings of the full trace, or you can individually fit the fast and slow decaying components. The fit interval is important and might require adjustments, since the number of available data points in the fast phase and the slow phase often is very different. The slow phase has much more data points, so you need to play around with the interval. That's pretty important. And if you want to fit the rise of the current, it might be even required to increase the sampling rate during the recording, because sometimes it can be really fast. What can also help related to exponential fittings is to investigate the dose dependence of the decay time. For SGLT, we found that the pre-steady state decay is dependent on the substrate concentration, but the decay of the transport phase is not. Next stop, reconstruction of transporter currents. This approach reflects mathematical processing of the data to replace the transport-related current decay phase with a steady-state current. Essentially, the procedure results in a current as it would be measured in conventional electrophysiology, which employs a constant driving force, achieving steady-state currents. Current reconstruction leaves pre-steady-state currents mostly unaffected, as shown in the right-hand example. Just the decay of the slowest current is removed and translated into a stationary current, so to say. But the point is, the whole process assumes that this component actually reflects transport. And in my opinion, the main advantage of this method is a better visualization of the transport phase. You can find the mathematical background of this process in literature described by Tadini et al. Here I just want to highlight one example, the nitrate transporter near C, the figure on the left. Here, the amplitudes triggered by nitrate and nitrate, the other thing, are pretty much similar in amplitude, right? But if you reconstruct the actual transport current, you can see that the pre-steady state of both signals are very much identical, but the transport current phase is halved for nitrate. Okay, method number 4, dose response curves. This is a very straightforward method, which we recommend to use in any case when new assays or target proteins are investigated. I tried to explain the situation with that ugly scheme here on the left. You can already see that plotting the peak or a later time point against the concentration will reveal different EC50 values. For example, at 1.2 seconds in time, the current is saturated already at 7 millimolar dose, the light green, while the peak still increases with 125 millimolar of the dose, the substrate dose, it's a dark blue one. Analyzing the integral will probably reveal an EC50 value somewhat in between. So different EC50 values for different readouts of the current indicate multiple current components with distinct kinetic properties. One example is SGLT1. The EC50 values obtained with peak current analysis differed for 1 millimeter and 3 millimeter sensors, and this is due to time resolution differences, which affect the relative contributions of the current components to the peak. In contrast, integral analysis is consistent across sensor types, and the EC50 reflects the transport Km found in literature. There are 2 other methods besides peak and integral. You can use reconstructed currents, as I just showed, also for the dose depends analysis, or you can use currents at specific time points, just any time point if you want. In the case of SGLT1, both match with transport Km and integral analysis, because all of those readouts are dominated by the transport phase. The time point analysis, however, was a little too random for us. Can you just use any random time point for EC50 analysis? So we had a closer look. We selected multiple time points in the current trace and analyzed dose responses for each individual time point. And indeed, obtained EC50 values were not random. The further away you get from the pre-steady state current phase, the EC50 values approach the Km for transport, because the further you go away, the less the current readout is affected by the pre-steady state. And for transporters which do not have a pre-steady state, we don't see huge time dependence at all. Last but not least, deconvolution. This is a method which identifies multiple peaks in a complex transient current. we are using a non-symmetric B-Gaussian peak model to mimic SSME currents. The process is very simple using Origin Pro, which has a built-in tool for this. So let's just go through it quickly. And as an example, we use GABA-induced binding currents, which are triphasic. So there are 3 individual current components, all coming from the target protein, from GAT1. First, you need to define the baseline. Then the tool will automatically detect the peaks, and we were surprised that I actually found the 3 peaks. And in the next step, it will apply a fitting using selected model. And here you might want to set some boundaries. Specifically, you want to say that all currents should start with the current rise when you actually perform the substrate jump. All currents are essentially triggered by the substrate, right? So we missed that in that example. We already have a more refined version. At the end, you will obtain the peak amplitudes and charge contributions of each individual current component, and that's very helpful to characterize the individual components. I hope these methods will help you identifying and actually finding out how many different current components you actually see, and also get an idea about their kinetic properties. But now we will look into potential molecular origins of the obtained currents. And we will start with having a look at what molecular processes can actually be electrogenic. There are 2 different processes, physically speaking, that can be measured as capacitive currents in SSME. One involves the change of voltage, and the other the change of capacitance. Let's look at the first term. Voltage changes are induced through charge movements. And, well, that could be ions moving into the protein's binding pocket, for example. But you actually don't need a charged substrate. Any substrate can induce conformational transitions within the protein that move charged amino acids. So protein movement, essentially, involving charges. And now it comes, you don't even need actual movement of charge. Imagine a fixed charge in the protein's binding site. Then the protein undergoes the conformational transition from outward to inward open. The ion was not moving, but alternating excess translocates charge, generating voltage. Coming to the second term, and this is much less obvious, but anything that changes the local or bulk capacitance within the membrane also generates a capacitive current. And there are also multiple examples. It could be like water displacement from a binding pocket resulting from substrate binding. Now that changes the dielectricity, and therefore it changes capacitance. That could be also a conformational change of the protein, which doesn't move necessarily charges, but maybe protein dipoles, for example, helix tilting. And also more related to artifact currents, that also involves phase transitions of the membrane by temperature changes, or the change of bulk dielectricity when compounds insert into the membrane. Just as a hint, in many electrogenic processes, both terms play a role and cannot be distinguished, but still the molecular origin of the recorded current can be determined. So in the next slides, I will show a few examples. Starting with the assignment of single pre-steady state events. A recent publication, also I think from last year it was, involves the measurements on the ATP-ADP exchanger. In absence of co-substrate, ATP and ADP concentration jumps generated negative and positive currents respectively. And those currents reflect the individual half cycles of the exchange process. So just ATP going in or just ATP going in because there's no nucleotide inside that can move out afterwards. So now you can imagine positive currents for ATP carrying 3 negative charges. How is that possible? And that's pretty easy. The explanation is more than 3 positive charges from the protein's binding site are translocated along, but less than 4, since ATP with one more negative charge generates a negative current. So based on the known properties of the nucleotides, the charge difference, the authors figured out the number of charges in the binding site. That's quite nice. And through mutational studies, those charges were even identified, attributed to individual amino acids, even the partial charge translocation. Another older study investigated the proton-coupled sugar transporter LAC-Y. Mutation of the proton binding site still showed the same pre-steady state currents as the wild-type protein under extreme conditions where the transport is completely blocked. Hence, the pre-steady state cannot be attributed to proton binding or proton movement or anything that has something to do with the proton, because, well, protonation is not happening anymore. Additionally, the rate of the pre-steady state current matched with the rate of sugar occlusion derived from fluorescence in that case. So complementary assays were used to compare the rates. And indeed, it was found out that the current we measured reflects the sugar occlusion process. Here comes the last example, the glutamate transporter EAT3. This one has a very complex mechanism, but it was known which substances are needed to actually trigger transport. In all cases, glutamate was used to trigger protein activity. With outward-directed potassium gradient and inward-directed sodium gradient, transport currents were measured. They were broad in shape. That's the very first signal here on the top. When potassium was absent, only a fast pre-steady state current was detected. And when sodium was also absent, glutamate didn't generate any currents. That means either glutamate is not able to bind to the empty carrier when sodium hasn't bound before, or glutamate binding is not electrogenic. Let's take it a step further. Multiple pre-steady state events could arise under different conditions, reflecting different molecular processes. One interesting example are ATPases. Here, a very old publication, I think, yeah, it could be even the very first publication showing SSME data. And it was about the sodium-potassium ATPase. It's known that ATPases are unidirectional transporters working as pumps. Accordingly, in presence of ATP, the upper figure, sodium jumps generated a fast positive current, but removal of sodium with the non-activating solution did not generate an off current. So the positive current, when providing sodium, must reflect a single pump event. Of course, these experiments have been carried out in absence of potassium, because otherwise we would have measured a stationary transport current. But what happens if you perform sodium jumps in absence of ATP? And that's the lower figure. You do see sodium binding to the pump, which is also electrogenic. And since sodium binding is reversible, you do see an off current as well when you remove the sodium with the non-activating solution. Another early publication is about the sodium sugar cotransporter MELB. The authors found that sodium binding is electrogenic, while sugar binding is not. Based on that, they developed a kinetic model and assigned the electrogenic steps accordingly. We also performed similar experiments for the human sodium sugar transporter SGLT1. So overall similar stoichiometry and substrate specificity. But interestingly, we found an opposite behavior compared to MEL-B. For SGLT1, sugar binding is electrogenic, sodium binding is not. While the overall transport model is the same, the electrogenic steps need to be assigned differently. I want to conclude my presentation with a few words on the molecular origin of electrogenic transport. And that's just a very general, to provide a very general idea. So potential major electrogenic steps in a transport cycle are typically those that translocate charge across a membrane. This can be the charged substrate, but also charged binding sites. For example, for sympoders, charged binding sites are often neutralized when substrates bind. But the relocation of the empty carrier translocates a charged binding site across the membrane, and that could be the major electrogenic contribution to the transport. Alternating excess of the charged binding sites are actually often measured in conventional electrophysiology when voltage is used as a trigger. So pre-steady state currents in conventional electrophysiology are often reflecting the relocation of charged binding sites without substrate. In contrast, SSME recordings more often detects events in the substrate loaded carrier as pre-steady states because, well, we use the substrate binding as a trigger. The existence of pre-steady state currents might help you with the assignment of transport electrogenicity, but only a little. If there is no fast pre-steady state, you know that the transport electrogenicity must occur in a step later in the transport cycle, beyond substrate binding and initial conformational transitions. So the first steps, the first fast steps up on substrate binding must be electroneutral if there is no fast pre-steady state. They are not detectable. If there is a fast pre-steady state, then think about the following. Consider that the transport electrogenicity could have the same molecular origin as the pre-steady state current. The two electrogenic phases, fast pre-steady state and slow transport, doesn't necessarily mean that there are two electrogenic processes that are different. The transport is just kinetically limited by the slowest step in the reaction cycle. And the existence of a fast pre-steady state just tells you that early steps in the reaction cycle are electrogenic. So that was a lot. And that's actually the last slide, because I wanted to conclude with the most simple scenario, and that is leak channels. So here you can see a current recorded from a leak channel. In that case, it's TMM175. Leak channels do not bind ions as transporters do. That means the electrogenicity can be attributed just to the passive diffusion of the ion used in the concentration jump. And well, that's it. It would be so nice if everything is so easy. Actually, I also wanted to spend some time on kinetic modeling, but sadly, I don't have time to dig into that. There are many examples in literature regarding kinetic modeling, and maybe this will make up for another webinar. However, I want to highlight one important thing. If you want to get a better understanding about your currents, use extreme experimental conditions. The more extreme conditions, so-called boundary conditions, are tested, the better you understand the origin of your currents and if there are multiple current components involved. And that's it. Now I'm ready to take questions, and thanks a lot for making it through.
Thank you, Andre. That was great, and a lot. There is more, as Andre already indicated, more we could share, more we could talk about that. So we're happy if you want to let us know your preferences here. Do you want to have a sequel or similar webinars in the future? We're happy to do that. So Andre, I have one first question for you. You finished with the simple case with the leak channel. So what's your gut feeling? And I know you're you're into numbers, you're into science, so that is an unfair question to ask. But what's your gut feeling about how frequently does it really happen that you only have one signal or one very, very dominant signal in your surfer traces?
Yeah, that's actually a very excellent question. We didn't prepare for that. No, I mean, if you would have asked me that maybe 10 years ago, then I would have answered, I think pre-steady state currents are more like special scenarios. Now I would say it's pretty common. Like I would even say it's like 50-50. The question is if the pre-steady state phase dominates or not, right? So if it's not dominating, then you can you mostly look at the transport anyway, and you don't need to look into separating these. So but you need to carefully look what's going on for each new assay, and then decide what analysis methods are the best fit for the readout, right? And as I stated, it makes sense to test a few methods because the eyes don't see everything. So it's helpful just to see if the EC50, for example, changes with the peak and the integral. That gives you a good indication already.
Thank you. Yeah, so there is one question here. Is there a way to distinguish if you have multiple pre-steady state signals?
Yes, so I think for that case, the convolution is the ideal situation. Multiple pre-steady state. I mean, of course, you can only distinguish currents if the kinetic properties are different enough, right? You can imagine you have a process, a conformational transition, a two-step conformational transition, which both steps are electrogenic, but the rate constant of both steps are, well, maybe only twofold different. And then it will be harder to distinguish those. But if the rate constant of the processes are different enough, then you can see different decaying phases, different exponentials in the decaying phases. And you can use the same approaches as I showed. You can use the exponential fittings. You can use deconvolution. That's what is helping. For the GUT1, for example, we saw 3 different reactions. We also identified 3 more reactions triggered by different substrates, like sodium binding, chloride binding. So we had some fun in applying all these methods to distinguish. You can do that.
There's a follow-up question if a 1 millimeter sensor would be enough. I think that would not change the answer. So of course it might be favorable, or the resolution gets higher, but it still needs to be different enough.
Yes, I mean 1 millimeter, 3 millimeter sensor difference. The key is that you can resolve faster currents, right? So if you have processes that are 10 milliseconds and 30 milliseconds, 3 millimeter sensor can resolve the difference nicely. If you have a difference between 1 millisecond and 5 milliseconds, then 3 millimeter sensors won't be sufficient because the time resolution is in the range of like 20 milliseconds, right? So it's about the range of speeds we are talking about.
Yeah, maybe you can go back to the slide with the deconvolution. So I think the question was related to that. There were 2 very fast peaks. So question is, are both of them actually pre-steady state, or do you know?
Yeah, that's a good point. I didn't want to make to open that box, but we believe that E1 is a pre-steady state current, and E2 and E3 are actual transport phases. E2 could correspond to a sodium leak triggered by the substrate binding, while E3 represents the coupled transport. But of course, we cannot be sure as of now, but we have indications, experimental indications for that.
Right. Thank you, Andre. Another follow-up question. When should you stop trusting? Well,
yeah. So that's actually also a very very important question, right? I mean, I would say if you see something once, never trust it. But if you can reproduce the distinct current features across multiple sensors, across multiple measurement days, maybe also with different purifications, and you don't see those features in controls, then you can trust this is something. But what it is, well, that remains to be figured out with also the help of complementary techniques.
Okay. So maybe one more question. So I'm working with Pendrin, an anion exchanger. Why does the addition of iodide, which is negatively charged, to the external medium generate an upward positive current in the SSM electrophysiology recordings? So why a positive artifact if the thing is negatively charged?
Mm-hmm. Yes. So I guess you added a salt in the activating solution, though there's also a positively charged ion. So you rarely just you cannot just use. I mean, you don't provide charge; you provide salts in general, and both salts are exchanged. Or if the cut ion is the same, then well, there are different possibilities. Is it? I mean, yeah, that I think that makes up for a discussion. I have questions.
Yeah.
So,
So please just contact Andre directly for in-depth discussion about the details. You are always very welcome to just contact Andre directly. If you want to get advised on data interpretation, if you're struggling with that, or if you want to have a discussion partner, competent discussion partner for this, just contact us, and yeah, we will follow up with that.
I would be very happy to do so. Yes, and also it's always interesting to see what customers and researchers do with the currents they obtain with recordings. That's of course I should have highlighted that it's not only that's not what we came up with. Most of these concepts and ideas have been introduced by customers and published. So we just try to share the different ideas to essentially help you in finding the ideal methods for your use case.
And we had one question, how to prevent contamination of the chamber with hydrophobic test compounds. Of chamber, I assume it's meant of the sensor well. That cannot be avoided. However, it's also not big of a deal, I would say, as sensors are usually single use. So if you use compounds like that, I would refrain from reusing the sensors or be very careful. Contamination, so if that relates to the SSM itself,
well, it changes the capacitive properties, and it can also change other artifact amplitudes by that contamination. So if you do sodium jumps in presence of compound and in absence of compound, theoretically, due to the change of capacitance, bulk capacitance, these artifacts can be affected. So it's important if you have aromatic residues to run controls also with compound to figure that out. And well, I would always add the compounds at the end of the measurement sequence, not before, because you can rarely wash them off.
All right. I think with that, we'll come to an end and close the session. So again, contact us if there are more questions coming up. We're happy to help.
So thank you, everyone, and we'll see you then in a different webinar. Bye.
All right.
Thank you.
Bye-bye.
Bye.